Let's go. Okay, I think people will still keep entering the webinar, but we will start. So first, we would like to welcome all of you today. Thank you for joining our webinar. We can say good morning to some and good afternoon to others. So this webinar will be about Excimer technology, and you will find out the what, the how, and the why. We'll have a quick look at the company. So who I am first, and then we'll look at Clartes, at the technology, and then I will hand over to Laurent. So my name is Anne-Maud Boyard, and I'm the marketing manager for Clartes. So basically, I'm responsible for all the marketing functions. I've begun my career in the Middle East. I've worked in Asia, in Europe, and in the Middle East, but I've also had the global functions. And I then moved to Europe, back to Europe. I hold 2 masters, one in international business and one in digital marketing. Moving on to Clartes, so who we are and what we've been doing so far. So Clartes was founded in 2014, and Exciplex was developed and came out in 2016. The company was ISO 13485 in 2016, and Exciplex got the CE mark. Then later on in 2017, we got FDA cleared, and today we're present in more than 50 countries globally. Moving on, we have already, as we said, we're present in more than 50 countries. So we have a broad global presence from US to Australia. Major countries like China, Japan is a big market for us. And we have already sold more than 700 devices globally. We basically have monochromatic Excimer lights. So we treat autoimmune skin conditions at a wavelength of 308 nanometers. There are 2 different types of phototherapy. You know, conventional phototherapy, where you expose the full body, and targeted phototherapy. So for us, with Exciplex, we are in targeted phototherapy, treating at 308 nanometer. Conventional is more UVA, BB, UVB, and narrowband UVB. We have a CE mark, as we've said. So our clinical indications are for vitiligo, psoriasis, alopecia areata, which is growing for us as an indication. We have mycosis fungoides and atopic dermatitis. So today we said we will focus on the more technical and scientific or aspects. So we have Laurent with us, who is a very experienced entrepreneur in the field of light-based devices for dermatology. He has a PhD in laser physics and a master's also in project management. Laurent was the founder of Dermoptics. It's a company that was later on sold to Quantel, and he worked for more than 10 years in product development. He's also managed product and R&D, basically from R&D to end users. Today, basically, Laurent is the CEO of Clartes, and he manages all the company's corporate culture. He provides us with a great vision of what we should all be doing and achieving as a team. He's in charge of all major, obviously, decisions, managing the overall operations and the resources. So most of us will actually report to Laurent. And he's the main communication point for us within the company. A great leader that we all enjoy spending our time with. So I will hand over to Laurent right now, who will tell you more about the technology. Thank you, Laurent.
Thank you very much, Anne-Maud, for this introduction. So as you said, I have many hats on me, and today I will take my scientist hat, and I will try to explain as far as I can what is an Excimer and how we do it in at Clartes. So the first question is, what is the difference between Excimer and Exciplex? We hear both names. And so basically the name Excimer comes from the molecule. A dimer is a molecule made of two atoms. And this molecule, in the case of an Excimer, only exists in the excited state. That's why Excimer basically is the contraction of excited dimer. I will explain this more deeply just after. For instance, the molecule made of two atoms of xenon, like a xenon-xenon, is a dimer, and then is an Excimer. In the case of an Exciplex, the molecule is made of two atoms, but the two atoms are different. So it's not the same like in a dimer. So it's a complex like a xenon chloride, for instance. So the Exciplex is a contraction of excited complex. So understand that in physics, from the point of view of scientists, all xenon chloride based devices are Exciplex and not Excimer. But in the usual language, I will use Excimer or Exciplex indifferently. So if I look at the energy, because everything remains in the energy curve of the molecule xenon chloride, which is the molecule that we use in the Exciplex and in all the 308 nanometer monochromatic Excimer light device. This molecule, you can see the two levels. So the ground level of energy and the excited level of energy of this molecule. So, and the bottom axis is the distance between the two molecules, so the xenon and the chloride. So, and it goes from 0 when you have a very strong molecule, and when they separate, it goes on the right, and you just have the xenon and the chloride completely separated. You don't have any molecule. And because of the slope of the ground state energy level, you see that because of the slope, the molecule that will be in this state will go in the lower energy level, of course, and will simply separate. So the xenon and the chloride the distance will grow and grow and grow because of the slope. And finally, you can now understand why it does not exist in the ground state, so only in the excited state. If I put a molecule in the energy level E0, like in the ground state, so we have the xenon and the chloride nearby, but I give this molecule an energy, then it will go in the excited state. And because of the curve of the excited state, after a while, the molecule will go, of course, in the lower energy level of this state. So basically, the xenon and the chloride will get closer and closer, and the distance will be always the same about this distance between the xenon and the chloride. After a certain while, of course, the molecule will go down in the ground state because it will not stay in the excited state. So it has to go back to the ground state, and this will be done by emitting a photon. And the energy of the photon is exactly the distance or the gap of energy that you have between the excited state and the ground level at this distance of between xenon and chloride. And what is very interesting is that because of this curve of the ground state and the excited state, you see that the distance between the two curves here is always the same. So the energy of the photon is always the same. And we will see why it is very interesting in the following slides. When the molecule went down in the ground state, then because of the slope of the ground state, it will separate again, and we will find again the xenon and the chloride separated. And we could do this cycle once again and more and more and more. as many times as we want, as long as we keep the xenon and the chloride. But how does the energy of the photon defines the wavelengths? There is a rule in physics that is very well known, because the light can be seen as photons and can be seen also as a wave, and there is a direct link between the photon, the energy and the wavelengths, and the relation is just a constant between them. And you see that if you multiply the wavelengths by 2, then the energy will be divided by 2. So the link is direct. And we've just seen before that all the photons have the same energy. The photon emitted by the molecule xenon chloride is always the same. So all the photons also have the same wavelengths. And if all the photons have the same wavelengths, then the global spectrum is narrowband. And this is the natural spectrum of the molecule xenon chloride. And this is what we can find completely naturally. So it is not filtered, it's just what we get. That's just physics. Now the question is, why 308 is the best wavelengths? So on this graph, there are 3 curves. So the first is the green one. It is the curve of efficiency of the light, depending on the wavelengths, to treat autoimmune skin disease. So it has been published by Parrish, something like 40 years ago, but it's still valid. It's how the light will kill the T cells. So it also works for all the autoimmune disease. We can see that the maximum is just below 300 nanometers. So if we want to be very effective to treat psoriasis, for instance, then we need the light, let's say, around 300. But the light is also responsible to of DNA break. And of course, it is something that we don't want to have in too large amount, because you know that it is linked to cancer. So in red, you will see what we could call the safety line. So usually it is measured in the opposite way. So we look the wave, depending on the wavelengths, which one will kill more the DNA, but it was more visible to put it like this. So here it's a safety line, so very safe, let's say safe in the UVA, and very safe, I would say, in the visible, and the more and more dangerous in the UVC range, of course, and even in the lower UVB range. And we see that at 300 nanometer, yes, the safety is very low. And what is interesting is that when we look where these 2 curves cross, they cross at 308 nanometer, and you have here in blue the spectrum that we just measured with the xenon chloride molecule. So this is a good thing that happened just, I would say, by nature, because all these points are only biology and physics. It's a good thing that the spectrum of the xenon chloride is just the best wavelengths, or I should say the best compromise between a high efficiency and a low risk. Now, how do we make an Excimer lamp? So you need xenon, of course, you need chloride, and you need something to put this gas inside. So it's basically it's a quartz bulb. After this, you need electrodes. So, and one very important thing is that the electrodes are outside the bulb. And this is very interesting, because when we will make the discharge between the 2 electrodes, this will the little atoms that will come out from the electrodes will not pollute the gas, and we will keep the very clean gas of xenon and chloride. And we've seen that as long as we have xenon and chloride only, then we can make a lot of cycle, and with a very long-lasting Excimer lamp. So we need the pulse generator. We make the pulse, then the molecule get excited, just as we've seen before, and then goes back to the ground level of energy by emitting a photon in all the direction randomly. Of course, if we make only one pulse, we will not make a lot of light. So what we do is make a pulse generator with a lot of discharge between the two electrodes. And at Clartes, we have a patent because we have developed the smallest electronic architecture in the world to make pulses of more than 7,000 volts. So it's a very high voltage. And we do these pulses more than 200,000 times per second. So 200,000 hertz. And of course, it has to comply with all medical regulation in the world. Because so on the left you have the spectrum, so we've seen the distribution, the spectral distribution, and concerning the spatial distribution, because all the photons are going in all the direction, we get a very homogeneous light in the output window, or I would say in all the area of the lamp. But with mirror, of course, we will get all the light in the output window, and you see that it's very I would say exactly the same level of energy that is given to the skin of the patient, whatever the place of the skin in the output window. And this is something very different from the lasers, of course, where you usually have a Gaussian shape with a very hot spot in the middle and a lower spot around. And this is a good introduction to see what is the difference between lamp and lasers in physics. Because, of course, I'm talking only about 308 nanometer Excimer light. We use in both cases the same gas, so it's xenon and chloride. We have the same pulse electrical generator, or I would say about the same. Sometimes it's little bit different, but the idea is still to make high voltage discharge at a very high rate. We have the same chemical reaction. It's xenon chloride. That's the physics, and the same photons are generated. So everything is the same, and of course, because we have the same photon, we have the same wavelengths. So that what is the equivalent in both lamp and lasers Excimer. No, what is different? So we also have in both cases the cavity with the pulse generator, and we have the electrodes. And that's the first main difference between the lamp on the left. As we've seen, the electrodes are outside the cavity, whereas in the laser, they are inside the cavity. And as I just explained, when we will make a discharge in the case of the laser, some parts of the electrodes, the metal electrodes, will go in the gas and will pollute the gas. And then you will have to change it after a while, maybe 1 month, 2 months, 3 months. And so it's a costly operation that you have to do very often. Another difference is that in the laser you have mirrors, so that it is resonant cavity, so that the photon will stay in the cavity and goes and back to before they can go out. So when I make a discharge in the case of the lamp, I get photon everywhere in all directions, whereas in the case of the laser, I will get all the same photons and all going in the same direction out of the through the output mirror. So in the end, what is the difference? In the case of the lamp, we will have only spontaneous emission in all direction, and it's a non-coherent light. In the case of the laser, we have stimulated emissions. So it means that all the photons are just a copy of themselves. And so they all go in the same way. It's a collimated beam with a coherent light. These two phenomena are very interesting in physics because you can focus a very large amount of energy on a very small area. So it can be very interesting if you need a very high amount of energy, if you want to burn something, if you want to cut. Lasers are very interesting in all fields, but in dermatology too. But in the case of autoimmune disease, we don't need that high level of energy. And of course, the lamp and the laser are very quite similar in terms of use and results. And that's what have been proven through articles that have been published. So I will not go through them, but just to say that there was studies that were done with a split body with a 308 nanometer Excimer laser versus lamp, and all the results showed a very similar results. So it is the same thing. But on the if we now look at the commercial product, the lamp versus the laser, it is this time very different. Because in the case of the laser, of course, it's much bigger because you have a lot of big things to put in, and that's the major point here of the laser. You can reach a very high level of energy and up to 150 milliwatt by square centimeter, which is very high. But the counterpoint is that you have only a small spot size. Just as I explained, you can focus the light, but then you have a small spot size. Of course, the price is very expensive. And as I said, because of the mirrors and because of the gas you have to change, the cost per year is very high. In the case of the lamp, it's much smaller, of course, and we can still get a very high level of energy, but limited, let's say, around 100 milliwatts by square centimeter. We can get higher in our labs, but commercially, we just limit the lamp to 100. But on a large spot size, something like 3 or 4 times bigger than the laser, with a price that is relatively affordable compared to lasers. And the good point is that you have no additional cost when you have it in your office. There are more than 200 clinical studies that have been published now to show that the monochromatic excimer light at 308 nanometer is very efficient to treat the autoimmune disease like the ones that Anmod presented in the introduction. So I will show you some before and after that we got with the Exciplex. Just a picture from around the world. So that was Dr. Shah on alopecia areata, a very nice result in a few sessions. Also, Dr. Adalia Rodriguez in Mexico, and once again with 5 sessions, it's an amazing result. Okay, that's another example. We also treat vitiligo, of course, in sessions. Sometimes vitiligo needs more sessions, like 32 here, and I'm sure that the pigmentation will be completed after another few sessions. That's an amazing result in 6 sessions only with Dr. Adalia. Dr. Gamal, nice result too. Psoriasis, of course. Another example of results on psoriasis with Dr. Shah. Palmoplantar psoriasis also is a very good indication. Sometimes you don't have any result with any other treatment, and you can try and you can put a very high level of energy and you get a very nice result on palmoplantar psoriasis. Atopic dermatitis also the light is efficient, even if it's not, of course, the first indication of this device. And same thing for the mycosis fungoides I would say, in the early stages. I thank you for your attention, and I will be happy to answer if you have any question.
We've received some questions, Laurent. Thank you all for your participation and joining us for questions. So there is one doctor that is asking if the protocol, what is the protocol for vitiligo and alopecia?
Oh, the protocol is most of the time always the same. We try to treat with the highest dose, the possible dose acceptable by the patients. So the protocol, we usually start with a low level because we don't want to have a side effect, and we slowly grow the dose session after session until the side effect. So it can be the redness, or it can be hyperpigmentation, in rare cases bubbles, but then we will decrease, of course, a little bit. But the best protocol is to go to this level. In the case of the vitiligo, we like to get this pinkness of the white skin. It means that there is a reaction, and then there will be a repigmentation after that. And same thing for the alopecia areata. We increase the dose and we stop when the side effects are no more acceptable for the patient. And the side effect, of course, highly depends on the country and on the doctor, on the patient. So it's the relation that has to be created between you and your patient.
Great, thank you, Laurent. Then we have another question from Dr. Shah, who's asking, can we use Exciplex in paintbrush mode for larger areas instead of stamp mode?
Yeah, of course you can do it. Some doctor are doing it, but first thing is that don't be too far from the skin, because as I told you, the light is going in all direction, and if you are 10 centimeters away, then the light density that you will get on the skin will be very low, and you will have to stay for 1 hour. So the idea is to keep very close, so the density is still around 100 milliwatts by square centimeter. But the advantage is that you will not see the sharp edges in case you get a little bit of hyperpigmentation. Then the edges will be smoother. And of course, if you want to treat a bigger area, you need to put more energy. So I would recommend maybe to put 4 times the level of energy that you would like to achieve, and to make an area that is 4 times the size of the spot size.
Great, thank you, Laurent. Then we have a question from Dr. Javier Ubogi, who's asking us, so he's saying good morning first, and he's asking for some unified dose schedule.
Hello, Javier. I know that you know better than me how to perform phototherapy, so I will not teach you many things in phototherapy. But I would be happy to share with you if you have new protocol. Many doctors are trying their own protocol, and so I cannot say that there is a unified dose schedule, but always the same. The strongest the dose you can put on your patient, the faster will come the results.
Yes. Another doctor is asking, what is the lifespan of the lamp, and does it need bulb replacement?
Okay, so we've seen that theoretically, the lifespan of the lamp is forever. But in reality, we cannot make the xenon chloride gas as pure as we would like, and sometimes part of the bulb, part of other gas are coming in the bulb, and then the power will slowly decrease. And but you should not change the lamp in the lifetime of your device. At Clartes, we provide a 3 years warranty for the lamp with unlimited use. So you can use it thousands of hours and during 3 years. And if never the power decreased, we will change it for free. But you should not change it. Of course, after many years, maybe the gas will get polluted, and then you will have to change it. But you should not.
Next question is from Dr. Fung from Australia, who is asking, with small device handheld, what is the risk of damage with handling or dropping?
I cannot say there is no risk, but in our experience, it doesn't happen often. As you say, at Mod, we've sold more than 700 units, and we are aware of 3-4 cases where the doctor or the nurse dropped the device, and a part of it had to be changed. Sometimes the bulb, sometimes just the output window. It depends on how it falls on the floor. But so it may happen, but not that often.
Yeah. Thank you, Laurent. Moving on to the next question we have with regards to alopecia areata, how effective is it?
By the way, it is very effective, and this is a growing indication for us, and I think for all the Excimer users. And we see more and more people, doctor treating children, because you don't want to make injection with children. So they just use the light, which is kind of a natural treatment with very nice result. And I think it will be a growing indication in the coming years.
Yeah, for sure. All right, the next one is, what is the age limit for the treatment? What age for the child, for example, would be acceptable?
There is no many study about it, and what I've read is that the limit around 5. I would not treat children below 5 because we never know what can happen at this age with their cells that are changing a lot. So I would not go below 5. That would, at least if I were a doctor, I would not treat below 5.
Thanks, Laurent. Next one is Exciplex effective in treating large areas of psoriasis, such as half body?
It is effective, and you can use it even 100% of the body, but I would say it makes no sense. If you have a very large area, like 50% of the body, I would recommend to go in a UV cabin for a while, because it will be maybe faster, and or you can take the anti-TNF alpha drugs, the biologics treatment, and after that, if a few percentage remains, then treat yourself with or treat your patients with the Exciplex, because if you have a half body, which is half a square meter, something like that, it will take a very long time. So I would not recommend treatment of body area more than 20%.
All right. Next one is, what is the dosage for patients who have used the cabins? Is it the same or a faster therapy? So you've kind of answered it already.
Yes, by the way, it's exactly I see the cabin and the Xiplex very complementary, because you will use the cabin for the maybe 10-20 first sessions, and when it has been quite well cleared, then you can complete the treatment with the Exiplex, like if it was the same treatment in the end. So you start with the big cabin and you finish with the Exiplex. Of course, if you don't have cabin, you can treat completely with the Exiplex. You can perform the treatment with the Exiplex.
Yes. Okay, 3 more. So we have a question with regards to experience in nails, for example, for psoriasis.
In nails, no, I don't know. I cannot answer this question, but it's interesting. I don't know any doctor who did that treatment, but I will ask. I don't know if someone in listening to this webinar has done some treatment, but I would be interested in this answer and if they could get good results. But I honestly, I don't know if the UV light at 308 nanometer can penetrate the nails.
We will
But
investigate them.
I think that there is a question about genitals and lids.
Yeah.
This, yes, in this case, no problem. You can treat and you can get very nice results.
Yes, so that was genitals and eyelids. And the last one is the results are visible after how many sessions?
As you could see in the before and after picture, it can be after one session sometimes, and sometimes it comes after a very long time. We've been discussing with Dr. Fenish in Tunisia, and it was sometimes going up to 3 months without seeing any results before he decided to stop. But what we usually recommend is that if you don't see anything after 10 sessions, the probability that the results will be great is low, and maybe you should stop. Of course, once again, it depends on you and your patient and the relation. And so you have to, if you still have hope, maybe you can continue. But the first result should come, I would say, usually in 3-4 sessions.
Yeah. Thank you. I think we've covered all the questions. If you have any other questions, you can email us at contact@clartes.com, and we will try to answer all your questions as soon as we can. Thanks a lot,
Thank you very
everyone. It
much.
was great having you, and have a great day.
Thank you. Bye-bye. And good day or good night, depending on you.
Yeah.
Ciao, ciao.